This is Unit 5B, Part 1. There are in the body three different types of muscle tissue whose function is contraction. They are skeletal muscle, cardiac muscle, and smooth muscle. And they're different from each other in their structure, location, function, and activation. And we're going to go through many of those. Most skeletal muscle is arranged in an unusual pattern of multinucleated fibers. So skeletal muscle is not called cells, but muscle fibers. So you have skeletal muscle fibers. Smooth muscle cells, on the other hand, are individual cells. They are also elongated, nowhere near as long as skeletal muscle fibers, but somewhat elongated. The contraction mechanism is dependent on two kinds of filaments. These are made of protein, and the two types of filaments inside the muscle tissue are actin is called the sarcolemma. The cytoplasm is called the sarcoplasm. Any term, in fact, that has myo or mys or sarco always refers to, at the beginning, prefixes to the words, the terms. All these refer to muscle tissue. We'll start with skeletal muscle tissue. It's packaged into skeletal muscles that attach to or cover the bones of the skeleton. The muscles are individual muscles. Skeletal muscle is considered an organ, so each individual muscle is an organ. The tissue at a microscopic level is characterized by stripes called striations. Skeletal muscle, of course, is voluntary tissue. It's consciously controlled. It can contract quite rapidly, but it tires easily, and it's responsible for motion of the body parts. Well, it's extremely adaptable. It increases in strength and size with use and decreases if it's not used. But it can actually recover fairly quickly from a period of disuse. There's a huge range in the amount of force that the muscle can contract and generate. And it can be very carefully controlled, consciously controlled, whether the person will exert just a few grams of force or quite a few kilos of force. Cardiac muscle tissue is only found in the heart. It is striped and striated, in other words, just like skeletal muscle. It is, of course, not consciously controlled. It's involuntary. It contracts typically at a fairly steady rate set by the pacemaker in the heart, the SA node. There are also, so it's internally triggered to contract, but there are also external neural controls that allow the heart to respond to changes when the body needs more or less activity in terms of pumping blood around. The third type, smooth muscle, is found in the walls of tubular visceral organs. Tubular hollow-shaped visceral organs like the stomach, the urinary bladder, respiratory passages, GI tract, blood vessels. These help force food forward or other substances through channels in the body. It is not striated, and it is, just like cardiac muscle, unconsciously controlled. It's under involuntary control. Now, the typical functional characteristics of all muscle tissue is that it's excitable or irritable. It shows irritability in that it will respond to stimulus. It is excitable. It's a effector tissue. It's contractile, so it can shorten. It's extensible. It can be stretched out or extended. And it has elastic properties in that it has some degree of recoil, so that it can resume its original resting length after being stretched or contracted. In terms of function, skeletal muscle is responsible for all body movement, posture, stabilizing joints, generating heat, and there's several functions. Cardiac muscle, of course, is responsible for moving blood, pumping it through the body. Smooth muscle helps mediates a number of different functions through the tubular structures that it surrounds, including helping move blood and helping maintain blood pressure, moving food through the GI tract, moving urine forward through the urinary tract, etc. So, as I said, each individual muscle made of skeletal muscle is a separate organ. It's made up of a collection of different tissues, which is the definition of an organ. There is the main tissue type found in it, muscle tissue, but you also find blood vessels, nerve fibers, connective tissue, adipose tissue, all coming together to form the organ, the individual muscle, the discrete organ. Here you see a diagram of a muscle with a tendon attaching it to the bone. There you have the tendon attaching the muscle to the bone. The muscle is arranged in a particular, skeletal muscle is arranged in within the organ, is arranged in a particular pattern. There is a sheath of connective tissue that organizes it into separate compartments. The entire muscle is covered by what's called an epimysium. So that's the external connective tissue sheath. And then within the muscle itself, you have fascicles. You have a number of different fascicles, and here you see one fascicle pulled outwards, telescoped outwards so that you can see it separately. And this fascicle is wrapped by a connective tissue layer called the perimysium. Okay, so each of these fascicles is wrapped with a perimysium. And then within each fascicle, you have several different muscle fibers. So each fascicle is a collection, a bundle of muscle fibers. Remember, there are no cells for skeletal muscle. They're arranged, they're not cells, they're fibers. They're multinucleated, very long multinucleated fibers. And each of these is surrounded by an endomysium. So going from the outside, the whole thing is surrounded by the epimysium, and that surrounds several fascicles that are each wrapped with or surrounded by a perimysium. And each fascicle contains a number of muscle fibers that are each of which are surrounded with an endomysium. So that's the arrangement of skeletal muscle as an organ itself. And they're described here, as I said before. You can go through that on your own. The nerve and blood supply is very characteristic. Each muscle is served by one nerve entering into it, bringing in motor neuron commands and carrying outwards sensory nerve commands. It is supplied with arterial blood by one artery, which branches out to smaller arteries than arterioles, and it is drained of blood by one or more veins. The individual skeletal muscle fibers in the fascicles are innervated with nerve endings that trigger contractions. And the contracting fibers, the contracting muscle fibers use a lot of energy, so they're always dependent on a continual supply of oxygen and nutrients from the arterial blood. Metabolic wastes like CO2 or waste metabolic products of breakdown of carbohydrates are all removed by the blood in the veins. The attachment, as I said, is by tendons, but you should understand that skeletal muscles, most of them, are attached over a joint. They'll cross over a joint and attach to bone in at least two ends, and they allow for movement at that particular joint. So for example, if you consider your biceps, it is attached to the bones in the lower arm, and that attachment point on the movable bone is called the insertion point, and it's also attached at the upper end to an immovable bone, which is your humerus, the upper arm, and that is the muscle origin. So each muscle typically is attached at either end, attached across the joint so that when it contracts it pulls on the bones in the lower, across on the other side of the joint, and that's the insertion, and its origin is on the immovable bone. They are attached directly through the fusion of the epimysium to the periosteum. I remind you the periosteum is a connective tissue layer around the bone. That's a direct connection, and they are also connected indirectly by a connective tissue structure called the tendon. And so once we start looking at the skeletal muscle fiber, what it is essentially is a very long cylindrical fusion product from many different cells. Using the word cell here is not really correct. It's a multinucleated fiber originally made of cells that fused together during embryonic development, and that product of the fusion is called a syncytium. The word syncytium refers to anything that's a fusion of several cells. The diameter of the skeletal muscle fiber can be between 10 to 100 microns. Just to give you an idea, a red blood cell is 8 microns in diameter. The key difference is that the length of it can be very, very long, up to hundreds of centimeters long. That would not be in a human, but in some animal that's quite a bit larger than a human. But the potential length of it is quite long. It's very many individual cells that fused together into a skeletal muscle syncytium, a fiber. The sarcoplasm is the term for the cytoplasm. The sarcolemma is the term for the plasma membrane of this skeletal muscle fiber. So the sarcoplasm typically contains glycogen granules, stored glycogen granules that are present as a source of carbohydrate for respiration, metabolic respiration, to be metabolized by the skeletal muscle fiber in order to generate energy. The other thing that it contains is a protein that can bind oxygen quite efficiently, and that's called myoglobin. In fact, myoglobin, just like hemoglobin, is red. And the reason meat that you see in the supermarket is red is because it is rich not so much in blood but in myoglobin. The fibers, of course, also contain many organelles, particularly mitochondria for generating energy, and there's, sarcoplasmic reticulum, etc. There are also these structures that I'll talk about later called T-tubules. It actually has quite a unique kind of, these skeletal muscle fibers have quite a unique kind of arrangement that we'll get into. The myofibrils are very densely packed rod-like elements that contract. They make up most of the volume of the muscle, in fact, and their arrangement is very uniform. So you get the series of banding of lighter and darker bands. I'll describe a bit later. What I didn't point out to you is that in individual skeletal muscle fibers, inside of each one you have many myofibrils, but we'll get to that. Here, what you see here is a single, actually what you see here is a section of a skeletal muscle fiber. It's surrounded by the, and remember, multiple skeletal muscle fibers are packed inside a fascicle. But when you get down to individual muscle fibers, you see that inside are many myofibrils. And here's a single myofibril, but there's many packed in there. The nuclei, remember, skeletal muscle fibers are multinucleated. They're formed by the fusion of many cells during embryonic development. And the nuclei are all pushed to the outer edge, so they're just beneath the skeletal muscle fiber cell membrane, which is called the sarcolemma. There are many mitochondria tucked in between the myofibrils, and there's this banding of dark and light stripes, as you can see here, alternating dark and light stripes that are present because of the arrangement of the actin and myosin fibers inside the myofibrils, which we'll show in a little bit. Now, when you look at a skeletal muscle cell, you see that it's packed with myofibrils, and the myofibrils themselves are made up of what are called myofilaments. These are long structures, insoluble protein structures that are made of contractile proteins that can slide past each other, overlap with each other as the muscle contracts. Now, the region in a repeated pattern in the myofibril of contractile bands and filaments sliding past each other is called the sarcomere, and I'll describe that to you in the next slide. What you see here is a section of a tubular myofibril filled with myofilaments. The myofilaments are two types. They're the thick thin filaments that's made up of the protein actin, and thick filaments made of the protein myosin. So if you look carefully here, you'll see that the blue ones are the thin filaments, and the red ones, the red strands, are the thick filaments. So you have thin filaments and thick filaments, or myofilaments, which extend the entire length, actually, of a region. Now, this area here, called the sarcomere, is the fundamental contractile unit in the muscle tissue. There's one sarcomere after another, all the way down the length of the myofibril. The thick filaments and thin filaments are arranged in a particular pattern. And if you look carefully, you can see that where they overlap is, in fact, the darker stripes or striations in a muscle, and where they don't overlap is and you only have thin filaments, that's where it's a lighter band in skeletal muscle. So it's a repetitive, carefully structured arrangement of filaments within myofibrils, within skeletal muscle fibers, within the muscle tissue, all arranged in parallel, so giving that striated appearance. The actual structure is described as the sarcomere. The actual structure described as the sarcomere is the minimal fundamental unit of the functional unit. It is bordered by two Z discs. These are sheets of proteins that anchor the thin filaments. So you have these Z discs, and they actually help connect myofibrils to one another. The thin filament is in what's called an I band. The Z line goes right through the middle of an I band, and on the other side, bordering on the other side, is the other I band. And in the middle you have the A band. And if you recall on the earlier slide showing the striations, the A band was the darker band striped, and the I bands are the lighter ones. What you see is that the thin filaments overlap with the thick filaments. If you look at one single region delineated by the Z discs, this sarcomere, the thin filaments are present in the I band area, and thick and thin filaments are present in the A band area. Right down the middle of the sarcomere is called the H zone. So you get this banding pattern. The H zone is somewhat lighter because there's no thin filaments. You have an M line down the center of that. So here you see it laid out a little simpler, in a simpler linear diagram here, which just shows 3 thick filaments and a few thin filaments. So here you have the Z disc, and there's another Z disc. That means that this entire area here is one sarcomere, and there's a thick filament made up of the protein myosin. So there's one, and there's another one, and there's a third one. and you have thin filaments also. Then that's made up of the protein actin. At the ends of the sarcomere, you have a particular type of protein which has elastic properties, and it is called titin. And these titin filaments actually are the largest protein in the body. They are, and that's why they're called titin. Titan is a huge god from Greek mythology. So you have titin associated with the Z discs, helping to attach thin and thick filaments, as you can see, and giving it elastic properties. This is the kind of way that the sarcomere, the fundamental unit of contraction within the skeletal muscle, is how it's described. The actual process involves sliding past each other of thick and thin filaments. The thick filaments, made of myosin, have multiple myosin molecules, each one having a tail and head structures. There's two smaller heads on it, and these are the structures, these myosin heads are the structures that will bind to the thin filaments. So you have two interwoven polypeptide chains making up these tails, ending in the heads, which are smaller polypeptide structures that can eventually form the cross bridging. The thin filaments, which are composed of the protein actin, each thin filament has a tropomyosin molecule or strand associated with it, and the tropomyosin, as you can see here, this strand there, I didn't put the right color here. These tropomyosin strands arch around the actin that makes up most of the thin filaments, and they have these troponin complexes associated with them. Troponin complex. What happens in skeletal muscle contraction is that these thin filaments and thick filaments will slide past each other so that the whole structure, the whole sarcomere, gets shorter. Okay, this is one sarcomere from this end to that end, or no, it's just a section of a sarcomere, I'm sorry. Now there's another structure that is important for you to understand, and this is at a somewhat larger scale, not quite as microscopic, and that is what is called the sarcoplasmic reticulum. It's quite a large smooth muscle, it's quite a large smooth endoplasmic reticulum associated with the myofilaments inside each myofibril. They run along the length of the myofibril and surround each one. And their function is in the regulation of the intracellular calcium levels. You also have these structures called T-tubules, which penetrate down into the interior of each skeletal muscle fiber, and they're penetrating in right where the A and I band junction occurs, and they're associated with the terminal cisternae. So it's easier when you look at it in this diagram to understand it. Here you see a section of a skeletal muscle, a single skeletal muscle fiber with many myofibrils inside. And then here, what you see in magnified, much larger, is a similar structure. What you have are what they've done here actually is just blown up this area here, this small number of myofibrils from inside of the skeletal muscle fiber. So we're just looking at these 5 myofibrils here, a short section of them. And the sarcomere, remember, goes from Z disc to Z disc. All right, and is made up of a band and half of the I band, right? And the M line, of course, is right down the middle of the sarcomere. The T-tubules are shown here in yellow, and they're running down from the exterior of the cell, and here you see a little bit of that skeletal muscle fiber, and here you see a short area of sarcolemma. So this clear structure all around is the sarcolemma, and we can see the bit just behind these 5 myofibrils. Right, there are 5 myofibrils here. 1, 2, 3, 4, 5. Right. So what we see is that there are these T-tubules that run down into it. They are associated with terminal cisternae of the sarcoplasmic reticulum. And then the sarcoplasmic reticulum goes out and forms these mesh-like network all over the myofibrils. So you have these tubules of sarcoplasmic reticulum. The cisternae of the sarcoplasmic reticulum contain calcium, and I'll describe the role there. There are also shown in this diagram quite a few mitochondria. The skeletal muscle fibers are full of mitochondria, generating the energy that's required for all the activity that they carry out. So the T-tubules are these elongated tubes that penetrate down right at the A-band-I band junction. They're associated with terminal cisternae and are continuous with the sarcolemma. They help to conduct depolarizing impulses down into the deepest part of the muscle. So what you have is a system that sends impulses which allow for the release of calcium from the terminal cisternae. The terminal cisternae will release that calcium, and it's the release of calcium that triggers contraction of the muscle. So when a muscle is receiving motor neuron signals from the motor neuron action potentials, impulses, those impulses will be transferred over to the skeletal muscle fiber and then dive down the T-tube into the T-tubules and trigger release of calcium from these terminal cisternae. So you have these T-tubules and the sarcoplasmic reticulum forming this system for linking the signal for muscle contraction, which originates with a motor neuron, and coming down from the surface of the skeletal muscle fiber, and triggering the sarcoplasmic reticulum to release calcium from the terminal cisternae. Well, what that triggers, in fact, is that the thin filaments are able to slide past the thin filaments so that they overlap. In a relaxed muscle, the thin and thin filaments are only slightly overlapped, but with the signal coming in for muscle contraction, what you get are that the myosin heads at the end of the thick filaments will bind to the actin, and then they slide past each other, the two types of filaments. So here you see a single sarcomere from Z line to Z line. This is an electron micrograph showing one Z line is here, the other Z line is here. So you have the A zone, or A band rather, and you have the I band here, and another one here with the Z line there and there. So one sarcomere covers this area here, that part right there. And you get a signal coming in which triggers release of calcium, and that triggers the thick filaments to interact with the thin filaments and cause a contraction. And you can see that they're sliding past each other, so that because they're sliding past each other, the I bands are getting smaller and smaller until actually in the electron micrograph you can see it actually disappears, because now what you have is wherever there is a thin filament, it is completely overlapping with the area of the thick filament, and so that I band actually on either side of the Z line essentially completely disappears. So this is a fully contracted sarcomere, and remember, there's one sarcomere after another on the left side and the right, going off towards the left and going off towards the right. It's a very long series of sarcomeres, so they're all sliding past each other. This is the model of this mechanism, movement or contraction, is called the sliding filament model. With release of calcium from the sarcoplasmic reticulum, each myosin head will bind and then detach multiple times as the thick and thin filaments slide past each other. It's kind of like a ratchet type of mechanism where the myosin head reaches up, binds, and then the two slide past each other, and then the myosin head releases, and then again binds further along, and then they slide past each other again. So it's a ratchet type of mechanism, and it occurs all along the muscle, along the myofilaments, within the myofibrils, within the skeletal muscle fibers, within the muscle, and the whole thing shortens. Well, in order for skeletal muscle to contract, it has to receive a signal, a nerve impulse from axons, from motor neurons. You know, the outgoing motor commands, and this of course is the somatic motor system that we're talking about, the somatic nervous system we're talking about, which sends signals to skeletal muscle, which is voluntary tissue. So you have nerve endings of the motor neurons come down, and they come very, very close, but not quite in contact, but extremely close to the skeletal muscle fiber. So if an impulse comes along, an action potential, it will trigger a response in the skeletal muscle fiber, resulting in an increase in intracellular calcium levels. which triggers contraction. This kind of linking of an electrical type signal that's traveling down the neuron and across a small gap to the skeletal muscle fiber and then down into the skeletal muscle fiber, triggering release of calcium and contraction, is called, that's the mechanism, is called excitation contraction coupling. When you have appropriate excitation, you will get contraction. So skeletal muscle fibers, as I said, are stimulated by signals from motor neurons of the somatic nervous system. The axons are present in the nerves that go down to the muscle fibers. Shouldn't say cells, should be skeletal muscle fibers always. They branch, the axons of the motor neurons branch widely, profusely as they go into a muscle, and each branch will form a junction with a single muscle fiber. And that junction is called the neuromuscular junction. That's an important term. Excitation contraction coupling is an important concept. The neuromuscular junction is an important term. And that's the end of Unit 5B, Part 1.